In this lesson, we will discuss the concept of the resting membrane potential, the electrical difference across the plasma membrane when a cell is at rest. In most mammalian cells, the inside of the cell is about minus 70 millivolts relative to the outside. This negativity is fundamental because it sets the stage for excitability in nerves and muscles, among others. Let's break this down into 5 different steps. Step 1. Ion gradients, sodium-potassium pump, proteins, and permeability. The first principle is ion gradients. Sodium is concentrated outside the cell, potassium inside. This gradient is maintained by the sodium-potassium ATPase pump, which expels 3 sodium ions for every 2 potassium ions protein. The pump is electrogenic, contributing directly to negativity inside, but more importantly, it establishes the gradients that drive diffusion potentials. Equally important is that the membrane is not equally permeable to all ions. At rest, it is far more permeable to potassium than to sodium. This difference in permeability means potassium largely determines the resting potential, which in most mammalian cells is typically around minus 70 millivolts. In addition, large intracellular proteins carry net negative charges at physiological pH. These proteins are impermeant and remain trapped inside the cell, while proteins also contain positive residues. Those are effectively neutralized by mobile anions. The negative charges, however, cannot be neutralized in the same way, so if you will, they count more toward the overall charge. Thus, proteins too contribute to the negativity of the resting membrane potential, but they are not the major contributor compared with potassium diffusion. Step 2. Diffusion potentials and selective permeability. Potassium tends to diffuse outward down its concentration gradient. As it leaves, it uncovers fixed negative charges inside the cell. This outward diffusion generates a diffusion potential. The balance point occurs when the electrical force pulling potassium back in equals the chemical force driving it out. This is described by the so-called Nernst equation. In its simplified form at body temperature, it looks like this. Here, 61 millivolts is a constant. Z is the charge of the ion, and the ratio compares concentrations outside versus inside. For potassium, this equilibrium potential is close to minus 90 millivolts. So, if the membrane were permeable only to potassium, the resting potential would equal this value. Step 3. Why potassium still flows outward at rest? Now, here is a subtle but important point. The actual resting potential of most cells is about minus 70 millivolts, not minus 90 millivolts. That means the membrane potential is slightly less negative than the potassium equilibrium potential. Because the cell is not quite at potassium's equilibrium, there is still a small driving force for potassium to leave the cell. In other words, at minus 70 millivolts, potassium is not perfectly balanced, so it continues to leak outward. This outward leak is counterbalanced by a small inward leak of sodium, because the resting potential is also far from sodium's equilibrium potential, which is about plus 60 millivolt. In the normal nerve fiber, the permeability of the membrane to potassium is about 100 times as great as its permeability to sodium. Though sodium channels do exist, they are few, and so typical animal cells are mainly permeable to potassium ions because of the presence of so-called lead channels. These potassium channels are always open, more or less always open, at almost all membrane potential ranges. They are not voltage gated. They have no natural gating ligands. They are unaffected by conventional potassium channel blockers. And about 15 families of them have been identified so far. The sodium-potassium pump then restores the gradients, preventing the leaks from dissipating them. This explains why potassium still tends to flow outward at the resting potential. The cell is closer to, but not exactly at, the potassium equilibrium potential. Step 4. The Goldman-Hodgkin-Katz equation. The actual resting potential reflects the combined influence of sodium, potassium, chloride, and other ions, weighted by their relative permeabilities. This is expressed by the so-called Goldman-Hodgkin-Katz equation. This equation includes the concentration gradients of the major ions, sodium, potassium, and chloride, and weights each by its relative permeability. Because potassium permeability dominates, the resting potential lies close to the potassium equilibrium potential, but sodium and chloride shift it slightly toward minus 70 millivolts. The last step, step 5. Thin layer of charge separation. It is important to note, again, that the resting potential does not mean the entire cytoplasm is negative. The bulk of intracellular and extracellular fluid remains electrically neutral. The potential arises from a very thin layer of ions lining the inner and outer surfaces of the plasma membrane. The lipid bilayer acts as a capacitor, storing opposite charges across its thickness. This microscopic separation of charge is what we measure at the resting membrane potential.